Erysipelothrix, Lactobacillus, Bacillus, Listeria, Gardnerella, and Corynebacteria. Here are your objectives for the lecture. For an overview, so we have types. We have aerobic or facultative bacteria. So we have spore forming as well. So Bacillus is a type of spore former. And non-spore forming, so Pseudomonas and Listeria are examples of that. For environmental presence, so these are commonly found in soil, water, air, and surfaces. These thrive in diverse environments due to their metabolic versatility. For growth characteristics, it grows well on non-selective agar media like nutrient agar or blood agar, and there is no need for specialized enrichment or selective conditions. Clinical and industrial relevance, so the pathogenic species can cause infections in immunocompromised individuals, and some examples would be Bacillus cereus and Listeria monocytogenes. For food poisoning, so some species can produce toxins that can cause gastrointestinal illness, and these are often linked to improper food storage or handling. For contaminants, so frequently isolated as lab or sample contaminants, and they may not be clinically significant, but they could interfere with results. All right, so we have Bacillus species. This is an aerobic gram-positive rod, and it is a spore former. The endospore can be centrally located, subterminally located, or terminally located. Most are motile, and most do ferment glucose, and most are also catalase positive. Bacillus species can be found in nature, such as soil, plant, and animal products. Bacillus can be a frequent contaminant. However, if you identify the Bacillus anthracis, which causes anthrax, or Bacillus cereus, which is usually from food poisoning, those are going to be considered pathogenic. Most Bacillus species are generally sensitive to tetracycline, aminoglycosides, and chloramphenicol. For Bacillus anthracis, this is an agent of anthrax. It's related to handling imported wool or goat hair, animal hide, or animal byproducts. So individuals who contract Bacillus anthracis due to exposure to cattle, which then can lead to the infection. Bacillus anthracis is also an agent of bioterrorism. So if you think back to 9/11 back in 2001, around that time frame, spores were sent out through the mail in the United States, and it caused about 18 cases. This is a facultative, large, non-motile, square-ended gram-positive bacillus, as you can see on the picture. Oftentimes it is described as a bamboo appearance because of the spore. So if you kind of look at the picture, you can see those little clearings, and that would be where the spore is located. In humans, anthrax has three forms, either a cutaneous anthrax, which results in a malignant pustule, which is the most common in the United States. The second is pulmonary anthrax, otherwise known as wool sorter's disease. And the third is gastrointestinal anthrax, which is the rarest, but it's also the most severe. Colonies are large, gray to white or white. They're non-hemolytic or weak hemolytic, and sometimes they're referred to as medusa head colonies, and the colonies are always going to be larger than like your staph or strep. Bacillus cereus, this is associated with food poisoning due to improper storage and handling of food. It is a gram-positive rod with a central spore. Colonies can be lavender in color with a slight green tinge to it that spreads kind of widely from the center. The colony is once again large compared to most other types of organisms that we've talked about already. And it is catalase positive and VP positive. Listeria species. Listeria is also found in the environment from water, soil, and animal feed. It is considered a saprophyte, which basically means that it lives on soil and also lives on decaying organic matter. A Listeria monocytogenes is the pathogenic, so when you find that one is always considered pathogenic. Listeria monocytogenes is a facultative anaerobe. It is a short gram-positive bacillus, and it is a non-spore former. It can grow at 4 degrees Celsius, so think about your refrigerator temperature. So that is actually one of the strategies for recovering this organism, is that you can put your plate in the refrigerator for it to grow. It is oxidase negative, but it is VP and methyl red positive. There is a wide variety of infections either associated with food processing, and food production. Can also be due to ingestion of contaminated foods. And also in pregnant women, Listeria monocytogenes can cause influenza-like illness and septicemia. Can also cause premature birth or premature abortion or stillbirth because this is one of the organisms that can cross the placenta. Listeria monocytogenes is usually diagnosed early from blood cultures. Neonates, if moms are infected and baby becomes infected during delivery, what you'll see generally is sepsis or meningitis. It is a reportable disease because it is usually caused from contaminated dairy products or cabbage. It does survive and multiply in the refrigerator. Colonies do resemble a group B hemolytic strep. However, Listeria monocytogenes is motile. It has end-over-end tumbling motility, so if you do a wet mount with the organism, you'll kind of see it tumbling around. It is catalase, VP, and methyl red, and CAMP positive. And next we have Erysipelothrix rhizopathiae. This is found in the environment in nature and can infect animals. So we become infected because of our exposure to those animals. It is a facultative, non-motile, non-spore former that is gram-positive. It grows on blood and chocolate augurs, along with PEA as long as it is with an increase of carbon dioxide. This will be a small, slender, gram-positive bacillus, maybe look a little filamentous. It is easily decolorized and can appear gram-negative though. So that is when you hopefully will get some notation from the ordering physician. So you will know to adjust your gram stain procedure to maybe let it sit a little bit longer for it to absorb and be able to show that it is actually gram-positive. So this is a catalase negative. It's hydrogen sulfide positive if you set it up on a TSI tube. In humans, it usually causes a self-limiting cellulitis on the fingers and hands, which is referred to as an erysipeloid. Infections can be from an open cut from the skin, and we'll usually see this in people who handle animals, so veterinarians and fish handlers. To recover the organism, we're going to incubate for 48 to 72 hours, so it is a little bit more of a slow grower than some other ones. It can grow as two different colony types, though. So it can grow as a small gray colony, which has a small, slender bacillus, or it can grow as a large, flat, rough colony with a filamentous bacillus. It's alpha hemolytic on blood agar, but it can also be detected by PCR. Lactobacillus is a gram-positive, non-spore forming facultative anaerobe. It is found in a healthy mouth, saliva, and lactobacillus can also be found in the female genital tract. It's catalase negative and non-motile. It does thrive in the acid pH of the vaginal tract, and it has a purpose there, so it actually helps to keep balance of the vaginal normal flora. Colonies will be a pinpoint, smooth, grayish-white colony, so it'll look kind of like a strep. It does grow better in increased carbon dioxide or anaerobic, and it is typically resistant to vancomycin. So Corynebacterium is a pleomorphic, non-motile, gram-positive rod with tapered or curved ends. Oftentimes it'll look clubbed or cocoon shapes or maybe even a V shape. These are oftentimes referred to as Chinese letters just because of the configuration. Your most likely species of Corynebacterium is the Corynebacterium diphtheria, which causes diphtheria. Diphtheria is contagious and is considered a febrile illness where we have an exotoxin that inhibits the protein synthesis. However, not all Corynebacterium diphtheriae produce a toxin. This is a facultative organism, non-motile, and is highly pleomorphic. So a lot of times they refer to as the Y or L forms because of the arrangement of the rods and how it looks on the gram stain. Diphtheria is considered an acute communicable disease with an upper respiratory tract infection and the toxin effects which can result in death. So we're going to culture it using our blood agar plate or a tellurite containing media. So that would be like something like a modified Tinsdale or Loeffler agar will be used to recover Corynebacterium as well. Something specifically about this organism to remember is that it does grow on Tinsdale and it will demonstrate a gray-brown halo around the colony. All Corynebacterium needs to be tested for toxin production, and we do that with a test called the ELEC test. And this is basically where you'll see a line of precipitation where the toxin and antitoxin meet. So that'll indicate a positive test result. Again, and there is more information about this in your textbook. And then we have Gardnerella vaginalis. This is a pleomorphic gram-negative or gram-variable coccobacillus and is considered normal flora of the vaginal tract of the majority of women. Cells will adhere to epithelial cells, which create clue cells or glitter cells. So think back to your urinalysis class. So if you do a direct wet mount from a vaginal prep, you may see these cells and know it's from Gardnerella vaginalis. Gardnerella vaginalis is sexually transmitted, and its primary cause of bacterial vaginosis. Women will present with a thin, milky white discharge with a notable foul odor. A lot of times we won't even culture this because if we see 20% clue cells and we can determine if the vaginal pH is 4.7 with that amine type odor, along with the physician's notation of the vaginal discharge, that's good enough for diagnosis, and they don't actually need to do a culture. However, Gardnerella vaginalis can be seen in other disease processes. So during postpartum, it can cause bacteremia or neonatal septicemia. So we would do a blood culture and plate it as soon as possible, especially if that specimen is drawn in a yellow top tube that has SPS, or the sodium polymethyl sulfonate, which can actually inhibit Gardnerella vaginalis. So if there's a clue that this may be your organism, you may want to draw directly into the blood culture tubes so it doesn't have that chance to be inhibited. Most of the time, Gardnerella vaginalis is treated with metronidazole and considered a universal treatment. Even though Gardnerella vaginalis may be recovered, performing an antimicrobial susceptibility is generally not routine because as of right now, it is treatable with metronidazole.